have noticed the clock on the wall. I think we should pay more attention on the clock on the wall from time to time. So it's my honor here to report my story to you, and not only to you, but to the professors that have been here in the past and that will come in the future. So the spirit theory of the human body is the focus of this work, and we use the embryonic development to produce some proofs that it is correct. Dr. Ivanova was supposed to present this presentation, but I had to replace her today. And we saw some reports in this congress that quoted on Liddell, Watson, and also Sunza, and I believe mine is more on the side of Sunza. Every time we build a theory, we have to test it with deductive and inductive methods, and every time we do experiment, we also need to prove a theory. However, in 2022, we have so many information that if we make a search or we try to find arguments on one thesis or another, we can provide an endless amount of arguments on this search. So it gets more and more important what would be the algorithm that we will use to sort this information. Otherwise, we might get lost or we might get confused. This is how we measure, and you can see the measurement of the information we have to invent new terms to describe the new amounts of information that we have. And according to the human body, this is very recent Human Cell Atlas project. There is 1 million types of human cells, and they are still counting in over 33 organs. And I don't believe I can list 1 million types of cells. And I can see now that we are counting molecules, and we also counting atoms with another project with the electromagnetic pulse. And this is very interesting. Another project that will other projects that have a very vast amount of information about our scope. This is the BRAIN 2013 project, and the European project also, which that was the same project for the United States. Now, the anthropology, we can have two approaches to the anthropology. We see two approaches, and the one is on the picture to the left, and the other one is on the picture to the right. And the first one is a picture from the Pittsburgh Zoo, and we can see here also an example of the negative and the positive space very well used in the design of this advertisement, so that we can find also the line, and we can also find the monkey. However, on this picture on the left, we cannot find the human. And the picture on the right, you can see this is a snowflake, and this is from the movie Frozen, and we can very well distinguish the human there. Now, this is another approach approachable to the human, where you can find it in the snowflake. The homology is a theory in the physics that we can, let's say, tells us that we can distinguish two different objects by the shape of their negative space, their opening. And the movie didn't play, but the movie was another advertisement for the use of the negative space. However, you can see the picture, and you can see the models over there, and some people even now believe that the DNA has emptiness in the middle. However, if we look at the picture of the DNA, we see that the empty space is not so empty as we believe. And it also counts with the other empty spaces that we find in the body. Now, this is a direct example of the influence of the negative space on the embryonic development. This is for the stem cells, and we can see that the electromagnetic stimuli change their properties. So either we have to isolate our research from the electromagnetic fields, or we have to use it on purpose. Otherwise, we're just ignoring what's happening there. Now, this is another example that's more close to us. We have all drawn these type of pictures when we study the nervous system and the cardiovascular system. And this is actually the fractal system that is also another theory in the physics. So the conclusion here is that we should collaborate more closely with the physics department, like we did with stars and all, and this will improve our theoretical understanding of the human body. The spiral theory of the human body was introduced on the Union of the Scientists last year. So we did some discussion there, it's still undergoing discussion. It has 12 major questions that give a little bit more different answers. And here now we use the embryology to prove that these answers are correct. What we used for materials and methods for this publication exactly is heuristic formation of gold, which is used in the Max Planck Institute of Human Development. We use the systematic thinking from the Schumacher Institute in Bristol, and also we use a holistic approach from the Jiangxi University of Traditional Chinese Medicine in China. And this is one normal ovary, and we know that the egg goes to the medulla for its development, and this is the whole pathway that the oocyte and the zygote will do during its development in the uterine life. And you can see it has a distinctive spiral pathway. Now this is how we used to draw our pictures for the gametes, the male gametes, and this is still some researchers and some tutors would still use this type of pictures. However, this turned out not to be very exact, and now we have a new model of the sperm cells, which is much asymmetric and spiral than we did before. This movie played, you can see here the movie number 2 is very, you can visualize very well the spiral movement, and we have a picture on the left side to show how the blast works from a spiral. It's not called spiral cleavage like with some animals. It's called rotational cleavage because the first and the second blastomer start to spiral in different directions. Otherwise, it's called spiral. And this is all controlled by the centromere, which you can see in the middle has a negative space. Now, this is the whole cleavage done in one picture, and even more. On the left side, you can see the zygote dividing into the blastomers. In the middle, we have everything that's happening in our life. And on the other side, there should be an analogical process leading to the last cell, which we have not yet determined. So here in the middle, we have all these 1 million type of cells, which is above human understanding, in my opinion. Now, here you have the Hayflick limit. This is the picture of the Hayflick limit, who is saying that the telomeres will limit the division of each stem cell or the zygote cell to 50 divisions. So at some point in our life, we calculate around 35 to 40 years. This will happen, and after this, we'll have the opposite pyramid, and we can see the body falling apart. Another aim for this theory was to bring together the understanding of the traditional medicine and the Western medicine, which is now going in parallel ways, which is not like either. And you can see here the local change, how we describe flesh and cultural life. What we see in the systems, in the human development of the embryo and the growth development of the human systems, you can see that the same pattern is repeating again. Now I will skip this one because the time won't allow me to show everything, but we can see that there is negative space above and below the embryo, and the embryo will become as a third part in this picture. And we can see the beginning of the macrospiral here with the many stages. This is the centrifugal macrospiral, And also during birth, we have all these pure processes there, like the umbilical cord and the birth process itself. And our discussion, I wanted to show you this picture. This is what the transformism demands, and they're trying to build artificial replicator. However, all the animals that are born this way does not survive so far, and I don't believe they will reach this goal more than they build colonial mass. Now, another concern is the in vitro procedures which are building new genes in our population. However, these genes are infertile, and we can see a very rapid change in the human fertility. Outwave is something that is very easy to understand in a physical way. However, in the human life, I don't think we have a full explanation of the outwave. Thank you for your time.